Okay. Hello, everyone. So, today we are going to talk about heredity, inheritance, and variation. So, welcome to this lesson or topic. So, where we will delve into the fascinating world of DNA and how it influences our traits and characteristics as a human. And throughout this journey, we will explore the structure and function of the DNA, which its processes and also the mechanism by which genetic information is passed down from your parent to you. So from one generation to the next generation. So that is the process of transferring or replicating the DNA from one genes to another or from one generation to another generation. Okay, so this is the first lesson of the chapter 7, which is the DNA and replication. So first is the introduction of DNA, then and next is the DNA replication. So first is, what is a DNA? So DNA is the deoxyribonucleic acid, or short is DNA. So this is the blueprint of life. So sa engineering pa, mo ni siya ang mga layout sa paghimo sa balay. So as a human, we also have a blueprint which we call the DNA. So it encodes our traits or our characteristics or genetic information that came from our parent, then our parent will pass it down to us. So, this complex molecule, it resides inside the nucleus. So, sa nucleus siya mismo na sulod. So, nasa sulod sa nucleus mismo, which is inside the cell. So, inside this nucleus, we have this mga genes or chromosomes which encodes or carries our traits from eye color to height. Like for example, your parent has a blonde hair, so naturally it will pass down to you. You will also have a blonde hair. So if your parents have blue eyes, green eyes, or any color of your eyes, or brown. So it will also pass down to you because it contains DNA, or it will be passed down from one generation to the next generation. So that is the GNA, how marvelous the DNA is. And also DNA replication. So this is the process by which we copy the DNA molecule, and it will ensure that this genetic information is passed down carefully or passed down faithfully from one generation to the next generation. So this is very vital for cell division and cell growth. So that is the DNA replication. So along this topic, we will going to discuss that these two major topics of this lesson. So, um, in your grade 9, di ba, na topic naman ni sa inyong grade 9, it was discussed. So, this topic will be expounded in connection sa DNA replication. So, replication is just a process by which deoxyribonucleic acid is copied identically. So from parent DNA to daughter DNA. So that's the cell replication lang siya. From the word itself na replicate. So ginakopy lang ang mga genetic information. Okay, so what are the learning objectives in our lesson 1? So first, you will going to understand the DNA structure. So by the end of this lesson, you will be able to describe the intricate structure of DNA. So it will also include its double helix shape. Sa DNA katong mag-ana siya. So that is the double helix and the component that make up its building blocks. So when we say building blocks, the foundation of the DNA itself, the structure. And the second you will be able to identify the replication enzymes. So, unsa man itong mga enzymes nga nakapadali sa paghimo or sa pagbungkag or pag-replicate sa ato ang DNA. So, you can identify the key enzymes that involve the DNA replication process. So, you can understand their specific roles. So, they have different roles in copying the genetic code or genetic information. And the third would be the completing base pairs. So inside the DNA, we have this nitrogenous bases. So these nitrogenous bases will have this power to replicate your DNA strand. So it will demonstrate your understanding of the complementary base pairing rules that govern the DNA replication process. Okay, so the structure of the deoxyribonucleic acid or the DNA. So first, let's have the building blocks. So what are the building blocks or unsa man jud ang foundation sa DNA? So DNA, it was composed of repeating units. So when we say repeating, nagabalik-balik lang nga unit. So same lang yun siya ang naadira but ginabalik-balik lang para maka-form siya og building blocks sa DNA. So these repeating units is called the nucleotides. So each nucleotide, it has three parts. So what are the three parts? First is the deoxyribose sugar or the sugar group. Next is the phosphate group and the nitrogenous bases. Again, the three parts of nucleotide inside the DNA is the deoxyribose sugar, the phosphate group, and the nitrogenous base. Okay, so where can we find the deoxyribose sugar? So hito. Mo ni siya ang deoxyribose sugar. So kaning color blue, letter S. Mo ni siya ang sugar group sa ato ang DNA. So what is the phosphate group? So kaning naka letter P. Okay, letter P ang phosphate group. Now, where can we find the nitrogenous bases? Mao ni siya ang nitrogenous bases sa ato ang DNA. So, again, we have three building blocks or three parts of nucleotide. First is the sugar group or the deoxyribose group. Next is the phosphate group. And the third is the nitrogenous bases. So we have four different nitrogenous bases inside our DNA. First is the adenine or letter A. Next is guanine, cytosine, and the thymine. Okay, so that is the four different nitrogenous bases. So this nani siya namoy makita nga 5 and nay kama sa taas tapos napoy 3 diri and nay kama diri sa taas. When we say 5 sa taas, that is ascending strand. So pataas siya. But if ang 5 naa sa ubos is we call it descending strand or pababa ang iyahang strand. So, for example, ang iyang oxygen sa iyang sugar here is nasa taas. So, maka-determine ta nga kani siya is ascending strand or naa sa 5 iyang strand. And if atong makita sa sugar group nga iyang oxygen is naa sa ubos, so that means that is descending strand or pababa siya nga strand. So that is mo na siya nakakompose sa double helix. So the same, repeating units lang siya, balik-balik lang siya nga units. Here, balik-balik, and also dito. Okay, so what are the pairs of these nitrogenous bases? Okay, adenine will always be paired with thymine. Guanine will be always paired with cytosine. Cytosine will be paired with guanine and thymine will be paired with adenine. So that is their pairs. That's why makakita taog letter A and letter T. So there mo ni sila ang pairs sa DNA. So double helix siya. And guanine paired with cytosine, thymine paired with adenine, cytosine paired with guanine. So this is our DNA molecule representation. Okay. So the DNA molecule that was arranged in a double helix structure. So mo ni siya. So double helix structure ni siya and it resemble a twisted nga ladder. So nakakita mong ladder pero balikos. So twist siya. So this is the two strand of DNA are held together by what band? Okay, the hydrogen bond. So hydrogen bond ang nagpa-connect sa ilaha. So we can see a hydrogen bond here kaning between sa two pairs of our nitrogenous bases. So hydrogen bonds ang nag-held together or nag-hold together with these two different helix or two different strand. Okay. Question. My question? Okay, I think none. All right, let's continue. Okay, so DNA nucleotides. So we have the first part is the deoxyribose, which is ang kaning sugar. Next is the phosphate group, mo ni siya. Phosphate group ni siya. And nitrogenous bases, which is kaning inyuhang makita. Okay. So, here, between here, unsa gani ang nagpa-bond sila or nag-hold together. So that is the hydrogen bond. Hydrogen bond ang unsa tawag na nag-hold together sa ilaha. So two different strand nag-hold together because of its hydrogen bonds. Okay. So, what is deoxyribose sugar? So, the deoxyribose sugar is a 5-carbon sugar that forms the backbone of the DNA molecule. So, when we say backbone, katong mga strand, gid mismo sa DNA. Dili itong murag ladder. So, ang ladder is mga nitrogenous bases siya. Pero ang katong murag haligi sa iyahang ladder is ang sugar or ang deoxyribose sugar. So, it is linked to the phosphate group. Ito, mo siya ang phosphate group and the nitrogenous bases. So, na-link siya sa duha, ang phosphate group as well as the nitrogenous bases. So, what is a phosphate group? So, phosphate group, it contains phosphorus. So, from the element of letter P, phosphorus. So, the phosphate group, this is a negatively charged molecule that provides structural support and connects nucleotide to form the DNA chain. So, diri niya ang kaning phosphate group, ang ihang purpose is to connect with the sugar or the ribose group or the pentose group. So, man siya ang iyang purpose here para mag-provide og chain or connection together para maka-form og DNA chain. So, the nitrogenous base, so these are the 4 molecules, the adenine, guanine, cytosine, or the thymine. So, these base are responsible for carrying genetic information within the DNA molecule. So, naa na diri ang inyuhang genetic information. So, sila ang nag-carry out kung unsa ang imuhang genetic information or genetic code para ma-determine kung unsa ang imuhang traits or unsa imuhang characteristics physiologically. So, sa outside or panlabas ninyo. So, dira ma-determine sa inyuhang nitrogenous bases. Question. Okay. Next, the double helix structure of the DNA. So, when we say base pairing na rules, naagi na siya yung rules, which is ang adenine or letter A, always gid siya, always pair with letter T or the thymine. It cannot be changed unless if naa siya sa RNA. But when we say DNA, its pair will be letter T or thymine. Thymine gid siya permente iyahang partner. So bawal siya mangita og lain pag naa siya sa DNA. And for guanine, always gid siya nga i-pair for cytosine and vice versa. Cytosine, iyang permente jud nga partner is ang guanine. Thymine, iyang fermenting partner is adenine. Adenine, ang fermenting pair is ang thymine and guanine is yung pair or always niyang pair is ang cytosine. So, dili siya pwedeng i-pair sa dili pwede i-pair ang adenine o guanine. Dili pwede i-pair ang thymine o cytosine. Why? Because they have different chemical structure. So, medyo complicated na siya if atuan na siyang i-delve gid sa ato ang lesson because it have a different structure na pod for that chemical bond. So, basta ang inyo lang yung i-keep in your mind that guanine will always be paired with cytosine and adenine will always be paired with thymine in the DNA. So, DNA. That is the base pairing rules. So, we also have anti-parallel orientation. So, di ba na may two-strand ang atoang DNA. So, two-strand. So, mo ni siya inyong mga nitrogenous bases. And here. So, this anti-parallel orientation, so nana siya 5 diri and 3 diri. Napo siya 3 diri and 5 diri. So, nga naman na siya. Because, important man ni siya for orientation. This orientation is very important for DNA replication and protein synthesis. Kay, dili ni siya magka-pair ang mga nitrogenous bases if wala siya anti-parallel na orientation. Kung 5 lang ni siya and also 3, tapos same gihapon 5 and 3, most probably your nitrogenous bases won't be paired with each other. So, dili siya magkapareho or dili siya makapares. Dili ni makita iyang kapares if same orientation lang siya. Dapat naa siya'y anti-parallel orientation. So, pag diriya gani ang 5 sa taas, this is ascending. But if ang 5 naa sa ubos, this is descending. So, ascending pag naa ang 5 sa taas and descending if ang 5 is naa sa ubos. So, these two strand of DNA run in opposite direction. So, with one strand oriented 5 to 3, here 5 to 3, and the other is 3 to 5. Okay. So, again, important ni siyang orientation for DNA replication and also the protein synthesis. Question. Okay, let's continue. Alright, so ninggawas na ang answer. Okay. Okay, so we have here, tanggalan na lang sa natatabunan na tuog. Saan man ipag-copy? Kaya na lang. Okay, let's copy this one. Copy ba ni? Ay, mali. Andra. Screenshot here. Okay. Okay, so we have here direction of 5 to 3, which is ascending. So we have nitrogenous bases of adenine, thymine, guanine, cytosine, adenine, thymine, adenine, adenine, thymine, guanine, guanine, cytosine, and adenine with the 5 to 3 direction or orientation. So to answer this one, we have here, since 5 man ang naguna and we have this anti-parallel orientation, so we must follow the rules. So 3, 3 ang first na to, followed by, since ang first letter is adenine, its pair will be thymine. And next letter is thymine, answer is adenine. For guanine, it's cytosine, cytosine paired with guanine, adenine paired with thymine, thymine paired with adenine, and these two adenines is paired with two thymines. Thymine is paired with adenine, guanine is paired with cytosine, so 2, and the cytosine will be paired with guanine, and adenine is paired with thymine. So its direction is 3 to 5. So this is descending na, descending siya na direction. So, ano siya. Okay, di na siya madako. Okay, so this is the pairs for this certain na bond. So this is the pairing of the certain nitrogenous bases with its anti-parallel orientation. So from 5 to 3 or ascending orientation, we have this descending orientation of 3 to 5 with the pairs of thymine, adenine, cytosine, guanine, thymine, adenine, thymine, thymine, adenine, cytosine, cytosine, guanine, thymine, and 3 to 5 orientation or descending orientation. Question? Any question? Okay, let's check your comprehension. All right, let's have here. Okay, so who can answer this in the blackboard? Who can answer in the blackboard? Let's check your comprehension. Okay, so the answer is 5 to 3 direction or ascending direction. So, okay, so thank you so much. All right, so I think great ng comprehension for this complementary strand of this template below. Okay, so let's have this DNA replication tayo. Okay, before we proceed with the DNA replication, let's have these enzymes that were involved in the DNA replication. So first we have the helicase. So ang helicase ang first na to nga enzymes. So when we say enzymes, they are the catalyst para mapadali ang pag-replicate sa DNA. Again, enzymes are like catalyst na magpadali or magpa-act fastly sa pag-replicate sa DNA. So first is the helicase. Second is the DNA polymerase, and the third is the DNA ligase. So, pag makakita mo mga letter A's, mga A's, that is mga example of enzymes. So, tanang na A's is mga enzymes. Nga na lang siya. Bantayin lang ninyo pag makita mo mga A's, that is enzymes. Okay, so let's determine what is the description for helicase. So, ang helicase, they are the one that unwinds the double helix of DNA. So, it will separate the two strands to provide access to the template sequences. So, later on, na ako'y picture or illustration that will allow us to understand the helicase. Second is the DNA polymerase. So it will synthesize the new DNA strands and using the existing strand as template, tapos siya ang mag-a-add og mga nucleotide one at a time. So this DNA polymerase will synthesize a new DNA strand. So na may mga parent DNA, ang katong mga stranded to is iyang pangbutngan og mga nucleotide one at a time para magkabuo og another DNA. So, ang third is DNA ligase. So, it will join the newly synthesized DNA fragment, creating a continuous strand of DNA. So, these are the concept DNA replication. Illustration in having the DNA replication. So, during the cell division here, muna ni siya ang cell division, so it will undergo DNA replication. So during the cell division, each cell will need its own copy of DNA. So this DNA replication is the process by which DNA makes copies of itself during cell division. So, DNA replication involves a series of steps which takes place with the help of certain enzymes. So, kay balo na mo sa mga enzymes. So, these enzymes are the molecules that will direct the certain chemical reactions. So, DNA replication begins through unzipping the DNA molecule through the enzyme helicase. So, moon siya ang pinaka-first na pinaka-first na enzyme nga mag-open sa atoang DNA. So, the DNA or the helicase, which is ihang gina-unzip, ang mura siyang zipper ang atong DNA, tapos ang atong murag katong ana dito sa zipper is ang helicase. So, this helicase will untwist the two strand apart by breaking the hydrogen bond. So, di ba, kani siya is ang nag-combine together or ang nag-hold together sa parent DNA is ang hydrogen bond. Meanwhile, ang kaning helicase is iyang gina-break ang mga bonds with this nitrogenous bases. So, this will create a replication fork or kani siya, Y siya og shape. So, letter Y. Yan siya. Replication fork or the Y shape. So, these two separated DNA strands will then serve as a template for the new complementary strand. So, kani siyang na-separate na strand sa DNA will become another strand or the new DNA strand. Okay. So, na siya, no? Iko na siya, gi-unzip na siya sa helicase. Tapos, ang pinakaunang complementary strand will be the leading strand. So ginatawag siya leading kay nag-una siya og ka-form og DNA. But meanwhile, ang kaning lagging strand is naiwit siya og pagkahimo sa DNA. So our new complementary strand is referred to as the leading strand. To initiate this synthesis of the leading strand, so we have here the RNA primer. So natay RNA primer, no? So it will produce by the enzyme nga primase. Okay. So ang RNA primer produce siya gikan ani. So gi-produce siya gikan sa primase. Tapos mo-attach na siya, attaches to the leading strand template. So this signals the start of DNA replication. So mo na ni siya. First ani is ang primase una. Then mo attach siya diri para makahimo siya og mga makahimo siya og mga primers or katong mga RNA primers. Pag moingon tag RNA kay one strand lang man na siya, no? So iyang gina-fill in ang void para maka-form siya og mga new strand of the DNA. So this signals the start of DNA replication. Tapos ang DNA polymerase 3 here ay mo ka-confuse ha kay ang kani sila is same lang ni sila og pamaagi para maghimo og new DNA strand. So ang DNA polymerase 3, it will then attaches beside the primer and continuously add the nucleotides. So ihang ginaadan og mga nucleotides here, mo ni siya ang nucleotide. So nag-a-add siya og nucleotide para mag-continue siya to create the 5 to 3 direction towards the replication fork. So understandable naman siya no nga. Basta kay ang DNA polymerase mo ni siya ang mag-attach diri sa primer, kani siya. Mo man siya ang primer, mo ni ang DNA polymerase. Ano no? Let me erase that one. Okay. So this is the DNA polymerase. Same here sa leading og lagging strand. Duha na sila kabuok. Kani siya is isa nila ang nabili na lang dire is ang DNA polymerase because leading strand siya hapit na siya mahuman. So dire sa lagging strand gipakita diri or gi-illustrate diri nga ang primer ang DNA polymerase, this is DNA polymerase ha. May makita. Let me. Okay. DNA polymerase. Okay. This DNA polymerase will attach beside the primer para mag-continuously add siya og mga nucleotide. This is nucleotides para maka-form siya og leading or lagging strand towards the replication fork. Sorry. So, the other complementary strand is referred to as the lagging strand. Here, mo na siya. Okay, so, so this again, leading strand and lagging strand. Okay. Time check. Okay, so once this DNA polymerase reaches this RNA primer, it detaches itself. So mag-detach na siya and it will the separate segments of DNA segments formed in the lagging strand are referred to as the Okazaki fragment. So kani siya. Mo ni siya mga Okazaki fragments. Okay, so after the Okazaki fragments complete the lagging strand, a cleanup is then followed. So, DNA polymerase 1. Ano siya? DNA polymerase 1 then replaces the RNA primers both in the leading and the lagging strand. So, since ang leading is okay naman siya, na-clean up naman siya. So, ang lagging is wala pa man siya nahuman og mga pang-add og mga nucleotides or I mean mga nucleobases, mga nucleotides bases. So, wala pa siya na clean up with this DNA polymerase 1. So, the strand are thereafter proofread before the enzyme ligase. So, this ligase will seal the gap of the DNA. So, tanang DNA is na siya mga gaps. So, like for this one, wala nagsumpay ang mga Okazaki fragments and that's why ang DNA ligase is padagan siya dito sa new strand of DNA para ipang-fill in ang mga gaps nga naa dito sa or naa dito sa new strand of DNA. Dapat walay gaps, dapat kumpleto tanan para buo ang imuhang new strand of DNA. So, nga na siya ka-complicated ang process sa DNA replication. Anyway, I will send this video para inyong mabalik-balik o paminaw. So, what really happens inside the DNA replication. Okay. So, we also have so, as can be seen, the resulting DNA helix is made of the resulting DNA helix R is made of from one old and one new DNA strand. So, kaning mga black nga DNA strand that is from the parental DNA. So, old strand siya, tapos ang kaning color gray, this is the new strand of the DNA. So, sumpay siya with that parental DNA to form a new strand of DNA. So, because of the result, DNA replication is referred to as a semi or semi-conservative process. Kaya wala siya tanggal or wala siya etchapera nga processes. So, tanan is ginakuha gid siya. So, conservative processes iyahang way. So, semi-conservative DNA replication. So, first is the unwinding. So, the double helix of DNA is unwound by helicase. So, mamuto yung pinaka-first, diba? Separating the two strands. So, unwound by helicase or iyang gi-unzip. Next is the base pairing. So, the DNA polymerase adds complementary nucleotides to each of the separated strand, thus creating two new strands. And 3 is the joining. So, ang DNA ligase, ihada yung join tong mga newly synthesized na fragments or DNA fragments forming two complete molecules. So, let me erase this one, ha? Okay. So, ang first na enzyme nga nagkuan para makahimo ta og replication is ang helicase. Followed by the ano nga yung next? DNA polymerase. So we have two different types of DNA polymerase, the polymerase 3 and polymerase 1. So the function of these two is it will add complementary nucleotides. So iyahang gina-addan og nucleotide ang mga parental strands. So diri iyang gina-addan og nucleotides here pod. Gina-addan niya og nucleotides ang DNA polymerase. Next is the ligase, DNA ligase. So the DNA ligase is iyang gina-join ang mga old, parental DNA and the new strand of the DNA, forming two new or forming two complete DNA molecules. So that is the semi-conservative DNA replication. It starts with a helix. Next is the DNA polymerase and the DNA ligase. So completing the nitrogenous bases of the of replicated DNA strand. So in DNA replication, each new strand is a combination of one original strand and one newly synthesized strand. So this one. Mo ni siya ang original, kaning naka-color black, and mo ni siya ang newly synthesized na DNA strand. Okay. So this is because the nitrogenous bases in DNA follow specific pairing rules. So again, adenine will always be paired with thymine, and guanine will always be paired with cytosine, and vice versa. So what are the key takeaways with our lessons today? So first, DNA structure. So DNA structure, this is a double helix molecule composed of nucleotides consisting of the first is the pentose sugar or the deoxyribose sugar. Next is the phosphate groups. Then the nitrogenous bases, which is four parts or um, four components or nitrogenous bases was composed of adenine, thymine, guanine, and cytosine. So another one is the replication process. So DNA replication is a semi-conservative, meaning each new DNA molecule contains one original strand and one newly synthesized strand. Okay, and another one is third key takeaways is that the key enzymes involved in replication include helicase, DNA polymerase, and DNA ligase. So the base pairing also, we can have this the specific pairing of base pairs, which is the adenine and thymine and guanine and cytosine and vice versa. So they are essential for accurate or faithful na DNA replication para makapass ta correctly sa ato ang mga uh, genetic information or genetic traits towards from our offspring, towards sa atong offspring. So from one generation to the next generation. Okay, question. Meron bang question? None? Okay. If none, so um I will give you an assignment in which you will answer this in your notebook. Answer in your science notebook. Direction would be identify the role of the following enzymes in DNA replication. Okay. Uh, One is DNA polymerase, two are helicase, three is ligase. Kana lang. Okay, these three. Okay, answer this in your science notebook. Deadline would be next meeting. Next meeting. Okay, so I think that's all for today, and thank you so much for listening. And I hope you have new learnings for this new lessons for today. Thank you so much, everyone, and have a nice day.